Hi, my name's Kyle, and we're going to show our model of our DNA with the Symphony gene right here that we're going to encode. We've got our +1 start site right here, downstream, upstream, and we're going to start off with our closed complex. And we've got our yellow coding strand with the 5' end, and the green template strand with the 3' end. And Whitney is going to start us off with the pre-initiation complex.
Thanks, Kyle. I'm Whitney, and this is MRI, and we'll be talking about the closed complex. The Symphony gene in eukaryotes is a class II gene that contains a root, fifth, and third cis-acting core promoter elements and cis-regulatory promoter regions. Initiation of the Symphony gene occurs in a sequence of specific steps. First is the formation of the closed complex. This involves a number of general transcription factors that bind to the core promoter region of the gene. Starting with percussion, tympani drum is the first to bind, with its root binding protein region contacting the root element. The cello comes in next to add depth and stabilize the interaction between tympani and DNA. The clarinet then binds to the third sequence on DNA to form a ternary complex with symphony. Once trumpet binds, it recruits the concertmaster to the core promoter to stabilize the pre-initiation complex. Lastly, piano and harp will be recruited to the PIC to complete its assembly. The large 10 subunit piano factor contains the kinase and helicase activities needed to melt the complementary DNA strands open and make the transition to the open complex. During assembly, it's important for the carboxy terminal domain of the concertmaster to remain dephosphorylated. The kinase activity of piano will later phosphorylate the carboxy terminal domain of the concertmaster to begin to scan for the DNA start site and induce a basal level of transcription.
Hey, I'm Dylan. So this is the proximal regulatory promoter region, and this is our tempo element, and we have the tempo binding factor 1, which will bind to it. When there are mutations to the tempo element, that is when transcription is reduced. We'll move to the distal promoter, where there are many elements affecting transcription. We've got the adagio element, which binds to, or the adagio binding factor binds to, that will repress transcription when it's bound. We've got the presto element, where presto binding factor binds, and that enhances transcription when bound. We've also got the bar element, where the bar binding factor binds, and when that happens, it helps to separate the symphony gene and keep it from being transcribed from neighboring genes. So it helps to block enhancer promoter interactions. And so all three of these elements comprise of the resonance region. The bar, presto, and adagio elements do. While each element and their factor differentially regulate the expression of the symphony gene, their collective activities direct the appropriate expression of symphony. Okay, so with the assistance of conductor interacting with tympani and piano and harp, the DNA is going to loop and bend, placing the general transcription factors in close proximity to the presto and adagio binding factors that enhance or repress transcription. And in this case, we have adagio binding factor bound, which represses transcription.
Alright, so we're going to have enhanced transcription via the presto binding factor, which is going to bind the presto distal element, and the conductor is going to mediate interaction between the presto binding factor and the general transcription factors in the pre-initiation complex. And for transcription to occur, we have to have a phosphorylated C-terminal domain of our concertmaster also via the conductor, bringing the piano with its kinase activity in close proximity with the C-terminal domain. Before elongation can occur, there are 3 stages of initiation that need to happen. After it's been phosphorylated, the helicase activity of piano melt and unwind the DNA to form the transcription bubble. The first stage of initiation that occurs is abortive initiation, which usually happens several times with less than 5 nucleotides synthesized due to the instability. And as they are synthesized, they are cleaved from the active site and lost. If abortive initiation gets to about 10 nucleotides, then promoter escape is favored and RNA synthesis can proceed. Productive initiation occurs after about 25 nucleotides have been synthesized. Concert master is fully and this signals the 5' end of the RNA to be capped via the capping enzyme. And that is the end of our initiation, and elongation can proceed to produce wonderful music.